Well, thank you for the opportunity to present today. So we're a visual industry, so I thought I'd start off with some before and afters. And so these are a couple of patients from a clinical trial that we ran that I'll talk about in a little bit more detail in a moment. These patients were treated with Daxxify, Revance's neurotoxin, in the glabella and the forehead. And I want you to focus not on the static lines, which are evident, but I want you to focus on two things. I want you to focus on the area between the static lines, and then I want you to focus on the reflectivity of the skin. So this is a before image. This is an after image. Another patient who had reasonably good skin to start with. Before image, look at the spaces between the static lines after treatment. Okay, so this is from intramuscular injection of Daxxify in the glabella and in the forehead lines. So just to remind you of Daxxify, for those who may not be familiar, just a quick intro. So Daxxify was approved in September 2022 in the US and subsequently approved in Australia and China. It's approved both for the aesthetic indication of glabellar lines, but also for the therapeutic indication of cervical dystonia. It comes from a highly purified core neurotoxin, 150 kilodalton core neurotoxin, and as you heard earlier, it's the only product that's formulated with a peptide excipient. That allows us to remove or not have human serum albumin or human blood source-derived albumin in the formulation, and the manufacturing process is animal protein-free. So this is a completely animal protein-free product. From a clinical standpoint, things that are relevant is it's room temperature prior to reconstitution. After reconstitution, it's stable for 72 hours. Other products are only stable for up to 24 hours, so we have 3 times longer stability in the fridge after reconstitution, which is a testament to the unique and differentiated formulation of Daxxify. Wholly US manufactured in California. Clinically, our data shows a fast onset. We've talked about, or it's been talked about, the extended duration. But then this other thing that we heard about in the clinical trials, and we heard after approval, was this benefit that people are seeing in skin quality. Some people are calling it the Daxxify glow. So, as we've talked about earlier, Dr. Sridharani talked about having levels of evidence and a bar for evidence, so we wanted to understand what's going on. So we took a two-pronged approach. We started looking at clinical trial. We did a small clinical trial using the best available technology to look at skin quality, and that's Canfield's Primos Visia CR clinical research system. So we did a clinical study, and we also looked at preclinical markers in the lab. So let me tell you a little bit about the clinical study. So 27 patients, obviously they acted as their own control before and after. And using the Visia CR system, we were able to look at 2 dimensions of skin texture. Macro texture, which would kind of correlate to the static lines that we saw on the patients in the previous photographs, and then micro texture, which is that fine texture of the skin that you saw between the lines. So we were able to quantify changes in skin roughness, both in macro texture and micro texture. And what we saw was a statistically significant improvements in both macro texture, which you might expect because toxins improve static lines, but also in micro texture. So you see the changes here. They look nice, but so what, right? These are just numbers. We also did patient-reported outcomes. So we used validated phase Q, and we got a variety of different patient-reported outcome measures on their skin quality. And what we did was then we correlated the change in skin texture with the patient's perception of improvements in skin quality. And what that allowed us to do for the first time was quantitate a clinically meaningful improvement, an objectively clinically meaningful improvement as relates to the patient experience. And you can see patients who were dissatisfied or strongly dissatisfied with their change from baseline, they didn't have a large change in their skin texture. Those who were satisfied or strongly satisfied had a relatively larger change in their skin texture, and you can see that here. So that allowed us to calculate the threshold. We identified a threshold of 0.14 millimeters for microtexture and 0.256 millimeters for macrotexture. And so when we took those thresholds, which are now the determination of a clinically meaningful improvement, and looked at the actual changes from baseline that patients had improvements in skin roughness, what you saw applying those thresholds was that between 2/3 and 70% of patients had a clinically meaningful improvement in both macrotexture and microtexture after intramuscular treatment with Daxxify. Okay, it's the first time this data, this has been reported, certainly for microtexture. So then we wanted to look in the lab and say, okay, what can be underlying this change in skin quality? So we look aged human fibroblasts in a culture dish. We added Daxxify. And we looked at markers of skin health. So we looked at a variety of collagen subtypes and elastin. And what we saw was that there were some dose-dependent increases at clinically relevant doses here. You can see 5 units per mil, 8 units per mil, and 10 units per mil of Daxxify. And you can see that there were dose-dependent increases in some of these collagen markers, and in particular elastin. And if you take a doubling of the mRNA expression, of the gene expression as being a clinically a relevant threshold, you can see that some of these markers hit that threshold for being a meaningful change in the expression of these markers. So Daxxify is a unique formulation. It's got this peptide in it. What if we just added the peptide to these fibroblasts? What would we see? And what you can see here is that when we just added the peptide, no neurotoxin, we saw, again, dose-dependent changes in a subset of these markers. Interestingly, some of the collagen subtypes, but in particular, elastin. So, interesting things happening with the peptide alone. The peptide is highly positively charged. Okay? We have different flavors of the peptide that have a predominant negative charge. So when we used a predominantly negatively charged peptide in the same environment, we saw no significant changes and certainly no dose dependence to any of the changes in these skin quality markers that we had studied. So there's clearly something going on with the peptide uniquely in the biology of how these fibroblasts are processing collagen and elastin and other extracellular matrix markers. We went to an ad board, presented some of this data, and people said, that's fine, but show us what Botox does. So fine, we did Botox, we looked at Botox, and these are the data for, again, clinically relevant or appropriate doses of Botox. And again, you see no dose-dependent changes, which you would want to see if you were looking at a truly biological effect. So yeah, to wrap up, we looked at protein. So gene expression is one thing, protein secretion is another thing. So in these fibroblasts, we looked at the secretion of elastin protein into the extracellular, into the microenvironment. Thank you. And what you can see is that, again, at clinically relevant doses, you see a twice the amount of elastin production in these fibroblasts that we saw with Botox at comparable doses, or indeed comparable neurotoxin amounts of Botox. And so with that, there's my summary. I'll finish on time. Thank you very much.